This is an application video about the benefits of the Edge-3D microscope for neuroscience research. Why would one want a three-dimensional microscope? The real reason is to acquire more accurate information, and in the process, to decrease misdiagnosis and increase productivity. Why do neuroscientists in particular need 3D? The real reason is that brain cells are complex three-dimensional structures, and they come together into very, very complex brains and nervous systems. Therefore, 3D technology is needed to accurately assess and understand the morphology of these complex structures. Here's an example of a single brain cell from the cerebellum of the brain looking with a conventional microscope. With a conventional microscope, you only see a small bit that is in focus at one time. But with the Edge-3D microscope, it's all put into focus so that you see from the top to the bottom. This is about 135 micrometers in depth. And in addition, you can see using the rotational 3D display system, you can see this very clearly, the foreground from the background. And if you happen to have a pair of three-dimensional glasses that are red and cyan, that creates an even more dramatic three-dimensional image. Here is again a three-dimensional image using the red cyan glasses. This is a single PreKG cell from the brain. It's about 100 microns deep, and one can see all the dendrites here, the cell body, and here's the axon. And with rotational 3D movie display, one can clearly see the foreground from the background and get even a more clear image of that nerve cell. In addition, the Edge-3D microscope has a fluorescence attachment, and this allows one to see, in this case, 300 microns deep into a cleared brain specimen that was fluorescently stained in order to see the projections of the nerve cells. All this is run by a very simple, what we call 3D panfocal software. It controls the lights, the cameras, and the focus of the microscope, and allows one to stack a series of images from the top to the bottom and put them all into focus. So this is the 3D stacking portion. Here's the focus. And then once it's stacked, you can see the image and view it with various different three-dimensional viewing options. The features of the three-dimensional Edge microscope are: it comes with the panfocal 3D software as well as a 3D computer that allows you to see the 3D with active glasses. It includes automated Z-focus stacking, which gives you extended depth of focus. And then you can view the images in 6 different forms of stereo 3D viewing. One is stereo 3D viewing, rotational 3D viewing, surface profiling, and other forms of 3D viewing. Thank you very much. For more information, please look at www.edge-3d.com.